In our next video lesson on membrane transport from Chapter 9, we want to look at gated channels. In previous lessons, we looked at porins and ion channels, both of which were always open. They only differed in regard to the measure of their selectivity. In the case of gated channels, however, they're not always open. They have two conformations, open and closed, as illustrated at the top of our slide. They respond to the change in conformation to some signal. It might be a change in pH or the binding of a ligand or small molecule. Our illustration here is the potassium channel from neuronal cells. It's voltage gated and it responds to depolarization. Let's see how that conformational change works. In our figure, you can see it is primarily alpha helical in structure. The gray and the blue shaded helices move across the length of that lipid bilayer, and we have red alpha helices at the base at an almost 90-degree angle. These function as it were a drawstring in a bag. On the closed conformation on the right, you can see those red alpha helices are close to one another. We've drawn the string tight, and that's forced the gray and blue helices together, thereby closing that pore. In response to some signal, those red alpha helices will move. They will separate from one another. We're opening the bag, and that allows the gray and blue helices to separate from one another, thereby opening the pore. In the case of the neuronal potassium channel, after it first opens, there's an N-terminal segment of the protein that moves to block the pore, and this is why it doesn't immediately reopen. Remember, that's important for making sure that our action potential moves in one direction down the length of that axon. Another example of a gated channel is a mechanosensitive channel, and we have that illustration on our slide. They open in response to membrane tension. Similar to the gated channels we just looked at, it's a set of alpha helices that move past each other to alter the packing arrangement, and again they have a closed and open conformation. On the top we have an illustration, a ribbon diagram of a small channel, and on the bottom we have one of a large channel. On the top illustration we have the closed conformation at the uppermost portion of our screen, and below that the open conformation. You'll notice in the open conformation we can see the size of the channel will allow the transport of the molecule. As those alpha helices move, however, we contract that pore to a smaller area. However, we can only move and compress those alpha helices so much. And so we would still, even in the closed conformation, have a pore that was sufficiently large to allow transport through that open area. In this case, the opening is lined with bulky hydrophobic residues pictured as the purple spheres, so that as those alpha helices move together to the closed arrangement, it's as if those bulky hydrophobic residues finish the job and complete the closing of that pore. The difference is even more striking in the large mechanosensitive channel on the lower right. A very large opening here as it contracts and those helices move, a very small pore. We've sufficiently closed our channel so that nothing gets through. Mechanosensitive channels control osmotic balance in bacteria, such as the ones we have illustrated here. Osmotic balance has to do with the difference in water concentration inside and outside the cell. These channels are also thought to be sensors for touch, hearing, and balance. Remember, they respond to differences in pressure. In our next video lesson, we want to look at other means of transporting molecules in and out of cells, and find out if all transporters move only one substance through at a time.